Hello everyone and welcome back to the 14th lecture of this NPTEL course on aquatic geochemistry. In our last lecture also we were discussing some of the water quality parameters and we are going to discuss more parameters in this lecture. So, we were discussing temperature and now we move on to turbidity. Okay, so, turbidity is a measure of water clarity. Okay. It describes the amount of light that gets scattered or blocked by particles that is floating in the water. So, here you will see there are two images. One is showing a very clean river, okay, and you can see what is there at the bottom of the water, you can see that, right. So, in this particular image the turbidity is very low as compared to this image. Here you see there is a lot of turbulence, okay. The water looks very muddy, it is very brown in color, okay. So, what is happening? There is a lot of sediments in this kind of water. So, the turbidity of this is high and the turbidity of this is low, right. So, this is just turbidity is just telling us whether it is clear, whether the water is very clear or not. Okay. Now, it is extremely an important parameter for aquatic life. Okay. And this turbidity, it is not only related to sediments, it can come from algae also. Okay. Many times you will see that the rivers will look green, the lakes will look green in color, that means it is full of algae. Okay. It is full of small tiny bacteria which is photosynthetic nature, green in color or blue green in color. So, they are changing the color of the water, or it can be full of weeds and you will see it is completely weeded, aquatic plants, large aquatic plants are there. So, that also creates a lot of or dead decaying organic matter. So, the water can start looking very dark, you can almost see it is very blackish in nature. So, that color and turbidity gives us an indication of how the water is, okay. And it can be of seasonal nature, okay. And turbidity can vary throughout the season. If it is in summer season the flow is very low, okay. When the flow is very low, it is water is very streamlined, and then there is no high flows are coming, it is very whatever, the water is very sluggishly flowing. So, the sediments will settle, the water will look more clearer, okay. But if it is very high discharge is happening, there is lot of turbulence in the stream, then there can be more sediment that is getting mobilized or getting mixed up, or there is more from the runoff. Okay, runoff meaning soil and other rocks and all that, they will start coming more into the rivers because they are coming with the flooded water, right. So, when they are coming into the system, they will make the river look very muddy and they will have a lot of floating waste, okay, a lot of leaves, a lot of small branches, small particles, and all that will be there in the water. So, that will cause the turbidity of the water to change, okay. Now, turbidity is best measured at site because otherwise you will not know whether it got settled down in the system itself. So, turbidity, temperature, these are measurements that we do in the field itself, okay. And because these are so physical in nature, we call them the physiochemical parameters. That means, they are mostly physical in nature, but they can also be due to the chemical reactions also. So, they are called the physio and chemical. So, physiochemical parameters. Okay. Now, so other parameters can also change the turbidity. For instance, human activities such as logging. Logging means cutting down of trees. Okay, if you are cutting down trees somewhere in the catchment, the soil will start eroding much faster. So, when there is a rain, the runoff will have higher soil particles, higher concentration of soil particles. So, the water will again start looking very sediment loaded. Mining, if there is a lot of mining activities going on, then that mine water, if it is released into the stream, that will be having a lot of rock powders. The entire atmosphere, in the entire air surrounding that mine may become extremely dusty. There will be dust settling down on the rivers, okay, on whatever surface water is there, on the soil, everywhere there will be dust. And if that is getting into the aquatic system, that will change the turbidity. Agriculture, so again agriculture, it will increase the soil erosion. Dredging, what is dredging? Dredging means, dredging is a type of fishing, you can say fishing, or due to mining also. So, fishing, sometimes the fishermen, they will throw the net. Certain kinds of fishes, they prefer the river bed, okay, they will stay near to the bed of the river. So, they will throw the nets and they will start, okay, they will start to scour the top layer of the river, okay, the river bed, the top layer of the river bed, and they will try to catch the fish, because the fishes will try to make a home in that river bed. Okay. So, then the sediments, okay, due to that dredging process, they will start getting the sediments that were settled, they will start getting mixed into the water. Okay. Otherwise, if there is a mining, sometimes gold and certain other precious minerals, they are found in certain rock types from which the river is flowing through, and these tiny particles, they are found in that river sediments. So, what these miners will do is, they will start collecting these bottom sediments. Since these minerals will be heavy, gold is heavy, it will start settling down with the sediments, right. So, they will start collecting these bottom sediments and they will filter out whatever is the extra not necessary things, and they will filter out and only take the costly mineral, and they will throw the unnecessary rest of the sediments. So, such activities can also cause changes in the turbidity of the water. Now, turbidity measurement is done using, very commonly using a sensor. Okay. It uses this principle of refraction, whether, so when you pass a light through this water, this particles, they will scatter the light, right. So, you have that sensor and that will measure how much light is getting scattered, okay, and that is measured in a unit called the nephelometric turbidity unit. Okay. So, a lower NTU value means the turbidity is low. If the turbidity, if the turbidity is very high, the range is very high. So, it can be from 100 NTU or sometimes 400, 500 NTU. So, depends on therefore, so that is the range of turbidity values. Now, why are we very trying to understand turbidity? Why, what is the importance of turbidity? See, turbidity can impact aquatic life, right. If the water is very muddy, if the water is having a lot of sediments and all that, then it is difficult for fishes to breathe, okay. Their gills will get blocked. If there is a lot of algae and all that growth, then what will happen, it will easily deplete the oxygen, then again the fishes will start dying, okay. All animals, all aquatic animals, they will start suffering due to lack of oxygen, okay. So, low light availability, low levels of oxygen, all this can cause fish death, okay. Also, due to algae, there can be certain types of toxins, okay, which can kill the fish. High turbidity can also make it difficult for the fish to see and catch its prey. Right. So, there are many carnivorous types of fishes, many types of carnivorous fish, snakes, birds, insects, they are all depending on their vision for catching their prey, right. So, that gets affected, then their reproduction through eggs, and they always bury their eggs in the sediments. So, that can get impacted. So, a lot of ways can be there in which the turbidity can impact the aquatic life. Okay. With this, next I come to another parameter, that is alkalinity. Okay. What is alkalinity? It is just opposite of acidity. Acidity measures the concentration of hydrogen ions. Alkalinity is measuring the bicarbonate and the carbonate ions. So, alkalinity measures the water's ability to maintain a stable pH level. It is also called the buffering capacity. Okay. Now, generally there are two types of alkalinity. One is simply alkalinity that we see, and it is also a measure of the total alkalinity. Total alkalinity is equal to sum of carbonates, okay, sum of carbonates, this one, then sum of bicarbonates, okay, and also hydroxides. Okay. So, that is my total alkalinity. And how we are going to measure it? We generally measure it with a titration techniques. Okay. So, you go on adding acid. Okay. So, here you can see the entire setup. Okay. So, with your sample, to your sample a certain indicator is added, a color indicator is added, and an acid of known concentration is going to be in this burette. Okay. So, you are trying to see when this sample will change its color. Okay. So, that will be your end point. So, that the acidity of this much acid is neutralized. Okay. So, whenever the color changes, you will note how much acid you took to neutralize this sample. So, you know the acid strength, and that should be equal to the alkalinity strength. Okay. So, from the acid strength you will know the alkalinity strength. Okay. So, we generally perform titration to understand what is the alkalinity. Okay. Now, alkalinity sometimes we can only measure bicarbonate and the carbonate values, and from pH we will get an understanding of what is our hydroxide concentration. Okay. Now, what gives rise to alkalinity? How is it controlled? Alkalinity is controlled mainly by the dissolution of carbonate minerals, okay, such as calcium carbonate, okay, magnesium carbonate. These minerals, they are very commonly found in sedimentary rock types, okay, sedimentary rock types such as limestone. Okay. This kind of rock types, they are made up of calcium and magnesium carbonates. Okay. So, they and they are very easily soluble. Carbonates are extremely soluble in nature. So, if water is there, these carbonate rocks, they will dissolve, and these carbonate and bicarbonate ions will be released into the water, that will control its alkalinity values. Okay. Now, even if the minerals are continuously reacting, another extremely important source of carbonate ions into aquatic systems is the dissolution of carbon dioxide from the atmosphere. In fact, I am not sure whether you all know or not, but I will say here that if you are looking at the processes, the natural processes of carbon dioxide removal from the atmosphere, you will see number 1 is photosynthesis, and number 2 is its dissolution in water bodies. So, all water bodies, okay, they take up carbon dioxide. Okay. So, first as I said is photosynthesis, and 2 is the water body. Now, since all inland water bodies, they have a lot of photosynthetic organisms, aquatic life is there, the amount of carbon dioxide that is entering is almost equal to the amount of carbon dioxide that is getting released also from that kind of water bodies. So, sometimes there is no net uptake, okay, sometimes there is, but generally in the inland water bodies the amount of carbon dioxide uptake from the atmosphere, where therefore it is acting as a net sink, is low. But for oceans, oceans is a massive reservoir where carbon dioxide is dissolving in the water and gets trapped and forms carbonate minerals, because there are certain types of organisms are also there. They take up calcium and carbonate ions and they start making shells, and these shells they act as a sink therefore. Okay. And all these limestones and all that, they start getting formed in the oceans, they are formed in the oceans. Okay. So, dissolution of carbon dioxide, okay, so CO2 in the atmosphere, when it gets absorbed, okay, it forms H2CO3. Okay, so this is H2CO3. This in its next steps gets break down into H plus ions and HCO3 minus ions. Okay. So, this is my bicarbonate ion formation, and if it again gets down to the other reaction, that is H plus and then CO3 2 minus is formed. So, my bicarbonate ion gets formed and my carbonate ion is formed. Okay. So, due to carbon dioxide dissolution in water. Okay. Humans also can sometimes change the alkalinity, because we are adding fertilizers, okay, we are adding rocks and all that to the soil, and from the soil it can change the pH of the water that is getting released, the agricultural waste water that is released into the natural aquatic system, that alkalinity can get changed. Okay. Another very important parameter, an extremely important parameter, is dissolved oxygen. Okay. Now, oxygen from the atmosphere dissolves in the water, and all fishes and all aquatic animals depends on this oxygen to breathe, right. Now, dissolved oxygen levels depends on many factors. First of all, whether the water is flowing or not. If the water is flowing, then there will be some turbulence, there will be some mixing happening, that water will have a higher dissolved oxygen concentration. Okay. Wherever there is a obstacle, wherever there is a wherever there is a waterfall, there is a nick point, there will be mixing zones with the oxygen in the atmosphere. So, at those particular locations you will see that the DO will always be high, but wherever the water is getting very slow, it is flowing very sluggishly, there the oxygen mixing with the atmosphere is controlled. Okay. It gets very little mixing with the atmosphere. Then the oxygen level depends only on the aquatic life. If there is a lot of aquatic life, it can quickly get the level can drop, or if there is a lot of decaying organic matter, okay, then that also can take up the oxygen, and you will soon see that the oxygen level has dipped. Okay. So, oxygen temperature is also another very important parameter. If the water is hot, the oxygen level will be very low. If the water is cold, it is at around 5 to 10 degrees or below, the water will have higher oxygen concentration as compared to hotter water. Okay. And depending on the water oxygen level in the water, it depends the fish can live or not. Okay. So, for instance, at a value between 9.5 to 14 milligram per liter, okay, dissolved oxygen is either measured in milligram per liter or percentage of saturation. Now, percentage of saturation varies from place to place. Okay. So, for that particular percentage of what is called for that particular location, you will always see what is the saturation level, and that percentage of saturation is decided based on that, or otherwise you can always report it in milligram per liter. Okay. So, when you see that the oxygen level is very high, okay, around more than 9.5, then all types of fishes can survive. Below below a certain level, you will see slowly the more sensitive fishes, they will not be able to survive. Only the very robust fishes which are rarely difficult to kill, those kind of fishes will be surviving in the low oxygen concentrations. So, this picture just gives you a glimpse of the different types of fishes that can survive in the different oxygen levels. So, if you find smaller fishes, you will know, okay, there is enough oxygen is there. Again, depends whether those small fishes are mostly of carnivorous nature or herbivorous nature. So, different species, a lot of differentiation is there. So, aquatic life is more number of species is there as compared to terrestrial life. So, there this kind of distinction is, you will see, is very complex, and there is no, you have to do a lot of studies to see, okay, which fish can indicate which. Okay. So, that is an entirely biological aspect, and if you are interested, you can go on looking that. Another important parameter is the oxygen reduction potential. So, the oxidation reduction potential is a measure whether that water is capable of oxidizing or reducing another substance. Okay. Now, this controls the ability of that particular aquatic system to either oxidize the substances or reduce the substances. Okay. That means, it is basically capable of cleaning itself, okay, of cleaning out all the waste products, right. So, if there is some decaying, okay, some organic matter is there, it will start quickly breaking it down. If the ORP is healthy, if the ORP is at a healthy stage, that waste product will quickly break down, get oxidized, and it will get removed from the system. It will settle down, and it will slowly it will get removed from the water body, it will settle down in the channel. But if the ORP is very low, that means that the water no longer has the power to clean or remove those decaying organic matter. Okay. So, that will remain into the system for a longer period of time. Okay. And ORP value does not only depend on the oxygen concentration, it definitely depends on the oxygen concentration. It also depends on the other constituents which are of redox nature. So, iron concentration, nitrate concentration, sulfur compounds, okay, all these can actually control the ORP values. Okay. So, in general, if the ORP value is high, okay, if the ORP value is not negative, then we say okay, the river is a healthy river, or the lake is good and it is clean and all that. Okay. But near the bottom, say suppose you have, this is a lake, okay, and you have water here, okay, but as you get down, okay, wherever there is this is, suppose this is the sediments, okay, this is the bottom of the lake, okay. So, here there is sediments, okay. Now, whenever this water, wherever there is interaction with the bottom water, here the ORP values will always be low, okay. Here the ORP values will always be low, because always it is interacting with the bottom sediments, okay, and it will start getting reduced, okay. So, even in a system, even in an inland system, there can be changes across the depth, okay. The ORP values can change across the depth, okay. And when the ORP and the dissolved oxygen values are very low, there will be lot of dead and decaying material in the water, and that cannot be cleared out or that cannot be decomposed, okay. So, ORP values are extremely important, because it influences the persistence of certain contaminants in the aquatic system, okay. Otherwise, if the ORP value was good, then it would have cleaned or broken it down, right. But due to the low ORP and the low dissolved oxygen, that system is that contaminant remains in that system, and therefore, its persistence can harm the aquatic life. Chlorophyll. Chlorophyll is another biological parameter that we measure, and chlorophyll is a type of pigment, right. So, chlorophyll A is what we are going to measure in the water, okay. So, whether the water is looking lightly green or very deep green. So, that you know how much is the chlorophyll content in the water, and that pigment is coming from all the green plants and all the algae and the cyanobacteria that is there in the water. And along with nutrients, especially phosphorus, chlorophyll is used to determine the tropic state of a water body. What is the tropic state of a water body? The tropic state of a water body describes the amount of biological productivity, okay. How much biological productivity is there? A lot of activity is there, no activity is there, that is found out, determined using the chlorophyll content and the nutrient content, especially the phosphorus, okay. So, based on the tropic state, we can either have oligotrophic, or we can have mesotrophic, or we can have eutrophic, or we can have hypereutrophic conditions, okay. So, you can see here the values are there, that the chlorophyll is less than 3 microgram per liter in oligotrophic, the phosphorus content is less than 15 microgram per liter, nitrogen is less than 400 microgram per liter, clarity is greater than 13 feet, okay. And almost the water looks bluish in nature. You can see swimming, you can do swimming, you can do rowing and all that you can do, okay. Now, in mesotrophic, again the chlorophyll content is a little higher, the phosphorus concentration also little higher, nitrogen, clarity reduces, there are more types of biological activity going on. Eutrophic is again more number of plants are there, more variety of fishes are there. Hypereutrophic is bigger fishes are there, bigger birds are there, okay. A lot of biological productivity is happening. The lake is looking absolutely green, certain sensitive kind of fishes, they will not be able to survive, the oxygen level can be slightly stressed. Okay, so that kind of system is a hypereutrophic system. Okay, and chlorophyll A is a very easy measurement. Sometimes we do it in remote sensely also you can do this kind of analysis, because again it is a light sensitive analysis. You can use remotely sensed you can use remote sensing techniques to generate the data. Okay, and we tend to see algae blooms, and if there is if there is a fish death event in a lake, we can see whether it was due to eutrophication, and whether the lake was eutrophic and hypereutrophic in nature, and then you can determine what was the actual cause of fish death on that particular for that particular event. And it is not the chlorophyll content that causes the fish death. So, the fish death is caused by this kind of cyanotoxins, and these cyanotoxins are released when the fish when the algae are dying and they are decaying, they release these kind of toxins, and they can be very detrimental for fish health. Okay. And if it is beyond an acceptable limit, the fishes are seen to die due to this kind of cyanotoxins. Another type of biological parameter that we measure is coliform bacteria. Coliform bacteria are a type of bacteria that is mainly found in the intestines of people and warm blooded animals, and it gets excreted in the feces. So, it is found in soil, it is found in water, and when we detect these coliform bacteria, we know that there is some kind of maybe sewage that is entering our aquatic system. There is some effluent mixing happening, there might be a pipe breakdown somewhere. So, beyond beyond a certain value, this fecal coliform cannot live. Okay. So, if you are seeing that the fecal coliform concentration is there consistently in that aquatic system, you will know that that system is getting a continuous supply from either effluent or sewage from domestic sources or agricultural sources, or from animal farms or somewhere, due to which that bacteria is getting inside the aquatic system. Okay. And if that water is again further used, okay, for drinking and for bathing, if people are bathing in that water, if people are using that water for drinking, then they can get affected, right. So, fecal coliform is absolutely should not be there in the natural water systems where people are using it as a drinking water or as for bathing water, right. So, when there is a certain when that water we know that it is used for drinking water, then we try to minimize our human interventions such that no fecal bacteria gets released into the water, because it will again come back to the food web. Another person who is drinking that water will get affected, right. So, diarrhea, as we have mentioned, all these viruses and this bacteria which gets transmitted through water is due to this kind of contamination, and fecal coliform, E. coli, all these are very common types of bacteria. So, we try to find out E. coli, because we know that if E. coli is there, many other types of bacteria can be there. Okay. So, suppose if there is a disease outbreak, okay, if there is a disease, if somebody is having a disease, okay, and through the feces that will pass out, right. Some of the bacteria and all that will pass out through the feces and the urine, and that if it mixes, okay, with a water that other people are using for drinking and bathing, and that it can get spread through water, right. So, therefore, we are going to, and every time it is not possible to measure that entire array of bacteria, entire array of viruses. So, what people generally do, we know that certain kinds are very specific. For instance, E. coli, fecal coliform, okay, these are coming definitely from all humans and animals, right. So, if we are measuring these two, then we know that if this is there in the water body, then a lot of other bacteria can also be coming from these sources. So, we say okay, now this water is gotten polluted from some kind of effluent discharge or wastewater discharge. So, we are going to take preventive steps now. We are going to see where it is going, where it is happening. We are going to recover that place, and we are going to take steps so that until and unless the water is now absolutely free of total coliform and E. coli, we are not going to allow any more drinking. We are not going to use it anymore for drinking purpose. So, this kind of E. coli analysis, total coliform analysis is regularly done, so to prevent such kind of disease outbreak and to prevent any kind of this thing. So, with this we come to the end of this lecture, and in the next lecture we are going to see some kinds of understand some key processes that controls the natural water compositions. I will give you an overview of all the aquatic systems, of the different types of aquatic systems, and then we will move on to the reaction based understanding of each system. So, in the next class we will see some overview of natural water composition for different aquatic systems. So, see you in next class. Thank you.